Now we're going to begin to talk about week 2. And in week 2 of embryonic development, we complete the tail end of implantation, but we also continue to see differentiation between the embryonic structures and the extra-embryonic structures like the placenta. The embryo itself will start to form some of the major body cavities, and we begin to see differentiation of different tissue types within both the embryo and the extra-embryonic structures. One of the first things that the embryo will do during week 2 is to develop into 2 separate layers, and then it's called a bilaminar embryonic disc because of the 2 layers. One layer is the epiblast that will go on to give rise to the embryo, and the second is the hypoblast, which is a more transient layer and is ultimately replaced by other cells. But that does give us our first body axis. So now within the embryo, we have a dorsal side, which is formed by the epiblast, and a ventral side, which is formed by the hypoblast, and that is our first axis. All of this occurs back on day 7 before implantation is fully completed. Now, during the completion of implantation, days 8 through 9, we start to see the formation of the embryonic body cavities, and that includes the amniotic cavity and the primary yolk sac. Now, with the amniotic cavity, we start to see fluid accumulating between the cells of the epiblast, so those are the cells in blue. Additionally, epiblast cells extend along the cytotrophoblast, forming a membrane that sort of encircles this cavity that's now called the amniotic cavity. The membrane is called the amnion, and we'll talk more about those in later lectures. The cells of the hypoblast proliferate and migrate to form a primary yolk sac, and the cells from the hypoblast and the primary yolk sac then fill the rest of the blastocyst cavity, so a cavity that existed from earlier stages of development, and they're now called the extraembryonic mesoderm. That first primary yolk sac is going to degrade, and in its place will form a definitive or secondary yolk sac. So we get a second wave of cells flowing out from the hypoblast that migrate to form the definitive yolk sac, or the secondary yolk sac, and that's ultimately where the primordial germ cells will form that we talked about a couple lectures back. Additionally, the yolk sac is a site of early blood cell formation, hematopoiesis. The next cavity we form is the chorionic cavity. So that extraembryonic mesoderm that was derived from the hypoblast cells divides into 2 layers, and the space in between them becomes the chorionic cavity. And that separates the embryo from the outer wall of the blastocyst. Now, up till this point, the embryo has been managing its nutrition and waste needs through diffusion from the mother system. So waste from the embryo diffuses into the mother system, and nutrition from the mother diffuses into the embryo system. But very quickly it becomes insufficient for the embryo's needs, and the process is taken over by a developing placenta. The placenta, as I'm sure most of you know, is a large support organ that supports the baby throughout the entirety of the pregnancy. It contains many blood vessels. The umbilical cord feeds into it, and actually vessels from the mother feed through the umbilical cord to the baby, and then vessels from the baby feed through the umbilical cord to the placenta. But, this is important, the blood vessels never mix, so maternal blood and fetal blood never mix. The blood vessels are very close to each other, but they don't join. Now, the placenta's job is to exchange nutrients and waste from mother to fetus and fetus to mother. There are actually 4 different tissue layers, and that layer of tissue, the placenta itself, actually forms a barrier that protects the baby from things that might exist in the maternal system that could harm the developing baby. Be it infections, toxins, medications, exposures, anything that might get from mother system into baby, the placenta can prevent most of that from getting through. And we'll talk later in the course about cases where it does not protect. So how does the placenta form? It starts with the syncytial trophoblast, and basically you start to get spaces that form between the syncytial trophoblast, and those are called the trophoblastic lacuna, or lacunae. Now, maternal capillaries, so blood vessels from the mother's system, grow into the syncytial trophoblast and they merge with these lacunae. Uh, After that, the cytotrophoblast, that's the darker green in these pictures, grows into the lacunae as well, and then the extraembryonic mesoderm grows into that cytotrophoblast, and they're called the primary chorionic stem villi, and once the mesoderm has grown into them, they become secondary chorionic stem villi. Ultimately, blood vessels will differentiate from the extraembryonic mesoderm, and they'll join with blood vessels from the embryo, and they'll create the sort of tertiary chorionic stem villi that ultimately are going to become critical components of the placenta, although all of that's going to take place a little bit later in development. But remember, the mother's blood system and the fetus's blood system do not merge. Now, by the end of week 2, we should have a bilaminar embryo with one body axis. We should see that the primary yolk sac has degraded with the secondary yolk sac in place. And the embryo is at this point suspended within the chorionic cavity on what's called the connecting stalk. So when you're looking at this picture, on the right-hand side, that pink layer on the outside, that's the endometrium, the lining of the uterus. And the embryo is contained within it, within this surrounding structure of syncytial trophoblast and cytotrophoblast, and then extending on this stalk of mesoderm into the cavity in the middle of the syncytial trophoblast and cytotrophoblast. The embryo there is that little blue and yellow structure. The development of the placenta and the circulatory system has begun at this point, although it has more work to do. Now, this is a good time to talk about the phenomenon of the molar pregnancy. There are two different types of moles, hydatidiform moles and partial hydatidiform moles. They're more common than you would think, particularly in very young women and women who are approaching menopause. The hydatidiform molar pregnancy does not actually produce a baby. There's no fetus. There's no developing body. What you get is a very large placenta with these lobular structures that make it almost look like a bunch of grapes. And you can actually see that appearance on an ultrasound. Now, the reason you get those lobes is that the fluid from the maternal circulation that's supposed to be sort of feeding into the fetus isn't, because there's no fetus for it to feed into, and it's accumulating in the placenta. Most of the time, these pregnancies will miscarry, but they can continue, and if they do, they make the mother really, really sick. So the HCG produced by the placenta is part of what produces the vomiting, the unpleasantness that goes along with the first trimester of pregnancy, and these pregnancies produce lots and lots of HCG. So these women really don't feel well. Now, the reason that you get this molar pregnancy, the hydatidiform mole, has to do with the chromosome complement. They are diploid, but all of the chromosomes have come from the father. And the idea there being that maybe these pregnancies form when dispermia occurs, so when two sperm manage to fertilize a single egg, but may then lose the maternal pronucleus, so the original nucleus from the oocyte, and then the two male pronuclei could fuse, and you have a diploid conception, but all of the chromosomes came from the father. And that becomes a problem because of imprinting. Basically, the idea that cells can tag their chromosomes to indicate which parent they came from, and that you use chromosomes from one parent or the other on occasion. So sometimes you're not actually using both. You're using one from one parent or the other, and it's very specific as to which parent you're using it from. So you need chromosomes from each parent in order to have a fully functional set of DNA. The idea of imprinting is fascinating. The students who are in the genetic counseling program will spend much more time on it. If you have questions or you're curious about it, I'm happy to chat about it, but it's more than we can cover in the course of this course. Now, to finish up hydatidiform molar pregnancies, they are actually at risk to form tumors and potentially cancerous tumors called choriocarcinomas. It's the blanket term for that is persistent trophoblastic disease. these are tumors that have to be treated as cancers with surgery and with chemotherapy. And once they've been treated, you follow the mother with blood testing for her HCG levels because those should drop as the molar pregnancy leaves her system. So once it's gone, you should see a big drop in her HCG levels, and you generally follow them until they are 0. The other type of molar pregnancy is the partial hydatidiform mole. In this one, you do actually get an embryo, but it's abnormal, and it's usually quite small with lots of birth defects. Usually it will miscarry, but on occasion these babies are born alive. They don't, however, live very long. These are triploid pregnancies. So again, usually dispermy, but in this case, the two male pronuclei and the female pronucleus all fuse together to create a triploid pregnancy. You do see persistent trophoblastic disease, but it's much less likely to result in choriocarcinoma. And you can follow these by following the mother's HCG levels once the pregnancy is ended to ensure that they go back down to 0 and to ensure, therefore, that the pregnancy truly is gone, that there's no tissue left from the pregnancy that might develop into cancer. These are really very random events, so a woman who's had a partial hydatidiform molar pregnancy, a triploid pregnancy, or a hydatidiform mole is very unlikely to have another one, although because we don't entirely understand why they happen in the first place, her risk is slightly increased.